Well, it's my pleasure to introduce Miranda Melen for the public seminar of her dissertation proposal defense. And for those of you who don't know, this is the final step in the process of advancing to candidacy, which is an exciting moment in the life of every PhD student. Miranda did her undergraduate degree at Santa Clara University, and she completed her master's degree in environmental studies at San Jose State University under the direction of Rachel O'Malley, who we are happy to have with us today, and also working with Justin Whittall. Miranda's thesis was on the mating system, reproductive ecology, and genetic structure of the Zyanthi sandhills endemic Erysimum terridifolium. And her work contributed to an important debate about the conservation of that rare species. After getting her master's, Miranda was a senior ecologist for the Habitats program at the San Francisco Bay Bird Observatory, where she worked to restore habitats that are home to sensitive and endangered species of plants and vertebrates. At the same time, Miranda taught as a lecturer at San Jose State University, teaching courses in restoration ecology and environmental studies, and developing her really prodigious talent in teaching. Miranda joined our lab in the fall of 2018 and rapidly became an essential member of our lab group and of the whole department. Working in partnership with postdoc Nikki Lustenhauer, Miranda helped write a successful major grant from USDA and has led a big team of undergraduates working on the evolutionary ecology of Dittrichia graveolens as part of that project. And she'll be talking about some of the work that she's done under that project, which will be her dissertation, but there's also a whole range of other things that she's involved in. In addition to doing outstanding research, Miranda is also an outstanding teacher. She contributes to the department in many other ways as well, leading the peer mentoring program among our graduate students, mentoring and fundraising for the Plant Science Club, and acting as our lab's webmaster, among many other wonderful contributions. So with that, I am pleased to turn it over to Miranda Melen.
Thank you so much. Welcome, everyone. Thank you for joining me this evening. I appreciate you coming to listen to my talk on a Friday evening. To begin, broadly speaking, I'm interested in species interactions at the confluence of native and non-native organisms. I have focused much of my scientific career on California ecosystems, previously exploring the perils facing endangered and at-risk species, as Ingrid talked about, as well as studying restoration of salt marsh ecosystems. In this PhD program, I've leveraged my past experience to explore how populations spread and how research and management can work together to find conservation solutions. Among the threats to global biodiversity, invasion from exotic species critically impacts the biodiversity of communities. Assessing risk of exotic species from an evolutionary ecology perspective can aid in management and eradication efforts. Exotic plants commonly spread along roads and initially achieve high abundance in disturbed habitats. Ruderal species have traits that aid in dispersal along roadsides and in tolerating disturbance. Life history theory predicts that because of inherent trade-offs, ruderal species will be poor competitors. However, with mounting evidence to support rapid evolution occurring in non-native species in their new environments, evolution could increase the ability of an exotic species to invade undisturbed communities. Studying invasions also provides novel insights into the fields of ecology and evolution. My work draws on several important ecological frameworks within the field. Here is my outline for today's talk, which will start with an introduction of my study system, followed by my research questions. I will then walk you through my research, which focuses on disturbance and biotic resistance, rapid evolution, competition and soil-mediated impacts, and seed banks and population persistence. I studied Dittrichia graveolens, which is also known as stinkwort. It is an annual in the Asteraceae family, and it readily germinates after rain events and then spends much of its early months as a small rosette. It stays in its rosette form until around June, when it begins to bolt and produces secondary growth. slowly taking the shape of a small Christmas tree and reducing the size of its leaves. Over the summer, it continues to grow until producing buds in late July, August, or September timeframe. In the fall, it flowers and has small yellow radiate petals. From about October through December, Dittrichia produces wind-dispersed fruits that easily come away on clothing, tires, fur, and they just float freely in the air. This species predominantly grows in disturbed soils and along roadsides, but it is starting to be found away from roadsides in California, which is concerning. For the purposes of this talk, I'm not going to be adhering to the strict sense of the word invasive that the government defines as a non-native species that impacts the health, economy, or the environment. Instead, I'm going to look at it from a broader definition of a species that is capable of moving into vegetation and successfully spreading. The reason why I'm choosing this form of the word is because the species I'm studying is so new to California and the US that little is known about its invasibility. Therefore, my aim is to dive into the process of invasion and use the term invasive here as a means of acknowledging that this species is able to move into vegetation, but its impacts are as of yet undetermined. Dittrichia hails from the Mediterranean basin in Europe and has recently expanded its northern range and has been found to flower earlier in those areas. It has been introduced to Australia, South America, and North America. In California, it was first observed in 1984 along the train tracks in Alviso. Since then, it has spread along transportation routes and has been identified in about 81% of our counties. The USDA has ranked Dittrichia as a species of high risk for invasion. The unsavory elements of this species include, but are not limited to, small barbs on the seeds that perforate livestock intestines, which causes enteritis and leads to death. The plant tissues have been found to taint milk after ingestion by some species. And this lovely photo from my friend is simply a snapshot of the contact dermatitis that can occur from getting the plant oils on skin that has been exposed. So far, I've explained that Dittrichia has late phenology that shifts its major growth to occur during summer months, when much of the vegetation in California is senescing. I've also explained that there is evidence that Dittrichia is capable of adapting quickly to environmental changes, since it has already adapted to flower earlier in its native northern range. It has been in California for at least 40 years and is found in more than 80% of our counties. It is a threat to livestock and can cause a nasty rash for humans. All of these issues culminate into serious concern about this species, necessitating continued research and risk assessment. Last summer, we surveyed 30 populations of Dittrichia in Santa Clara County. We looked at paired roadside and off-road sites and found that there was no relationship between bare ground and Dittrichia population size, and that Dittrichia cover was actually higher in more diverse off-road sites. The findings from this preliminary study were counter to what we expected and led to some really interesting questions for an experimental approach for understanding whether Dittrichia can move away from roadsides and invade plant communities. Which leads us to my research questions. In my first section, I will ask, how do disturbance and biotic resistance from competitors affect Dittrichia's ability to invade established vegetation? In section 2, I will ask if rapid evolution has contributed to the invasion success of Dittrichia populations as it spreads away from roadsides. In section 3, I will ask whether Dittrichia alters the soil in a way that reduces the recruitment of other species. And in my final section, I will ask, how do Dittrichia seed germination, dormancy, and viability affect the longevity of the Dittrichia seed bank? And is dormancy reduced in populations from the range edge? Disturbance is a critical process in many ecosystems, providing heterogeneity and influencing diversity at both the patch level and landscape scale. However, it can be a double-edged sword because disturbance also reduces competition by freeing resources, which colonizing species, including exotics, can exploit. Roadsides, transportation corridors, and other disturbed environments facilitate primary spread of exotic species. The movement of species away from roadside edges and into established vegetation is a critical phase in the invasion process, and land that's adjacent to roadsides could easily be a stepping stone for invasions by providing natural experiments for adaptation and rapid evolution. Ruderal species are associated with these disturbances and are often the first to colonize bare ground. But the presumed trade-off is that those species are rarely good competitors. In previous research, we found that Dittrichia was able to establish populations away from roads, leading to questions about roles that disturbance and biotic resistance may play in influencing invasion. In my first section, I will have two experimental components. The first experiment will look at the response to disturbance, competition, and biotic resistance in a field setting. And the second experiment will complement the fieldwork by exploring the nuances of competition using a microcosm approach. With the help of friends, families, students, practitioners, and lab mates, I have established a field site at Blue Oak Ranch Reserve in Santa Clara County, which is a former rangeland. Here, each plot has 16 Dittrichia with a random treatment applied in a block design. Here's a photo I took just last month in April, placed next to a conceptual map of the treatments with a call out showing the gridded Dittrichia plants. There are 4 disturbance treatments: control, low, intermediate, and high. Remember how Dittrichia is a ruderal species found mainly along roadsides? Well, I hypothesize that Dittrichia will do best in high disturbance plots because they reduce competition with other plants more than the other treatments. And with increasing competition through reduced disturbance, I expect Dittrichia will not grow as well. In the low disturbance treatment, I removed last year's thatch, which can create cross-generational impact on plant communities. In the intermediate disturbance treatment, I removed only the above ground biomass to reduce competition. And then in those high disturbance treatments, I removed both the above ground and below ground competition. One possible outcome from this experiment is that with increasing intensity of disturbance, Dittrichia will benefit. This pattern could occur if Dittrichia is most limited by competition and therefore relegated to roadsides. But the species could quickly move away from roadsides if competition is reduced. The design of this study will tell us more about the mechanisms of competition and how Dittrichia responds. If competition is important here, there might be ways to increase competition or to make communities more resistant to invasion. Biotic resistance occurs when resident species in a community prevent or limit the ability of exotic species from invading. The limiting similarity hypothesis proposes that biotic resistance results from direct competition between functionally similar species. Researchers have long posited that competition is more intense between species that share traits, including close relatives. Therefore, a functionally similar species may limit the ability of invaders by increasing direct competition and decreasing available resources. Here again, we see that photo from April, adjacent to my conceptual map, with the plots that are now highlighted in orange as locations where a functionally similar tarweed was seeded. I selected the native tarweed, Hemizonia conjusta, subspecies Luzilifolia, as a species that has similar phenology and is found at Blue Oak Ranch Reserve, growing in the grassy areas. When comparing this treatment to control plots, I hypothesized that Dittrichia will do poorly when the native tarweed is present, because I expect that that tarweed will be better adapted to the grassland conditions and obtaining resources than Dittrichia. I expect that in plots where Hemizonia establishes, it will reduce the ability for Dittrichia to grow as vigorously because it is that functionally similar species that may do a better job of that direct competition with Dittrichia during those summer months when growth activity is really robust for Dittrichia, but resources are depleted. To complement my field experiment and to test stages of development that are pretty challenging to have in the field when you're ethically trying to work with an invasive species, I'm going to be using a microcosm approach. And then I will be looking at invasion in established communities in a greenhouse experiment. The design of this experiment is still forming, and I welcome suggestions from anyone with microcosm experience to comment at the end. Rapid evolution is concerning in the study of exotic species because it adds uncertainty to risk assessment and the prediction of invasions. Colonizers found along roadsides are expected to show adaptations to disturbance and to a degree of physical stress, but they prefer conditions of low interspecific competition. As rural exotics transition from actively spreading to residence in less disturbed communities, natural selection may act to favor traits for rapid resource acquisition. Dittrichia is a ruderal species, which is expected to perform poorly in competitive environments. However, Dittrichia in its native range has been shown to evolve rapidly. Therefore, evolution of competitive ability may allow Dittrichia to invade established vegetation away from roadsides in California. Here I focus on whether we are seeing differences in Dittrichia populations that would signal the presence of adaptation. Remember the field experiment at Blue Oak Ranch Reserve? Well, this experiment layers a genotype concept onto that experiment by comparing seed that have been collected from paired roadside and off-road populations. The second experiment here looks at competition in a greenhouse setting to see if there is any evidence of evolution of competitive ability. Last November, we identified 16 populations of paired roadside and off-road off-road locations in Santa Clara County. We selected sites by identifying off-road populations of Dittrichia and then locating the nearest roadside population, which was the presumed source. These populations were used in the Blue Oak Ranch Reserve experiment with the different levels of disturbance. At each plot, there are Dittrichia representatives from 8 roadside sites and 8 off-road sites. This shows biomass of roadside and off-road populations that were grown in a common garden. The 1-to-1 line shows no evolution. Points will fall above the line if the off-road populations have some improved performance relative to their roadside pair. I predict that in control plots, off-road populations will have an advantage over their roadside pair and have higher biomass. I predict that as competition increases, Dittrichia sourced from off-road populations will show evolution of competitive ability and will have better success than roadside populations. In high disturbance plots that best replicate the competition found along roadsides, I predict that the roadside and off-road pairs will be fairly well matched and therefore fall along that 1-to-1 line. For my next experiment, I will use a bioassay to assess differences between roadside and off-road populations, focusing on competitive ability. I intend to use a non-native grass as a challenge species in this experiment. I predict that Dittrichia will have higher biomass when growing alone compared to in competition with challenge species. I also predict that off-road genotypes will be less suppressed by a competitor than genotypes collected from roadsides. If I see off-road genotypes performing better than roadside genotypes, this could indicate evolution of competitive ability in roadside genotypes. The impact that a novel species will have on its recipient community will depend in part on its resource use and competitive ability. In addition to direct competitive effects, plants can influence the growth of co-occurring species indirectly through their effects on soil characteristics, both abiotic and biotic. Explain the impacts of an invasive plant, both through direct plant-plant competition as well as through soil-mediated effects, can have important implications for management. In my third section, I look at soil-mediated impacts to assess differences between invaded and uninvaded soils. There's literature that supports Dittrichia having some allelopathic chemicals, but I wondered if looking at the bigger picture of soil-mediated impacts, if that would provide more insight into patterns I have seen in the field and how Dittrichia gets a foothold in new communities. I will conduct a bioassay comparing 4 soil types. I will collect soil samples from roadside sites throughout Santa Clara County, and at each site, I will identify a pair of invaded and uninvaded patches within about 20 meters of each other. I'll collect invaded soils from around the roots of living Dittrichia, and uninvaded soils will be collected from a similar depth and size of hole 20 meters away. I will also collect extra uninvaded soil to amend with dried leaf material from Dittrichia and dried leaf material from native species. That will create the amended types of soils. I will then grow native species in each of these soil types. I predict that native species grown in invaded soils will have smaller biomass than those grown in uninvaded soils. I also expect that native species grown in Dittrichia amended soils will be smaller than those grown in native amended soils. Both of these patterns would indicate that Dittrichia has altered the soil conditions in ways to reduce the growth of other species, which would mean that removing Dittrichia from a site needs to include the remediation of the soil. Seed banks are a critical phase of the plant life cycle and affect population dynamics and the maintenance of plant diversity. Long-lived seed banks are an important part of the life history of many weedy species. They are therefore a critical contributor to a recurring seed source of invasive species. In colonizing populations, we can also predict that dormancy may be selected against in order to propel spread. In a review by Funk et al., they found that seed bank research was severely understudied compared to the level of desire from resource managers, thereby highlighting an inherent mismatch in research focus and management needs. So we know that long-term germination behavior is not often studied and is actually quite challenging to study, which is probably why it is so poorly understood despite its ecological and evolutionary importance. Little is known about Dittrichia's seed bank. However, in discussions with land managers, questions about Dittrichia's seed persistence often arise. Previous research by Brownsey et al. Tested optimal temperature and light for germination in a greenhouse, finding that there's a wide range of temperatures and light that are conducive to Dittrichia germination. Observations that they made of Dittrichia germination in the field suggest that primary dormancy may be non-existent due to the high rate of sprouting after rain events, especially in the first year. Yet, to date, there have been no field studies to quantify patterns of dormancy for Dittrichia. So in this section, I will conduct 2 complementary experiments that will feed into a holistic look at Dittrichia's seed bank. In 2018, we collected seeds from Dittrichia populations in Santa Clara County, or the core region, where it was first identified and presumed to be a resident the longest. We also collected populations from the Sierra Nevada foothills. I like to call that the invasion front, where those observations have been more recently documented and presumed invading into those areas more recently. Seeds were grown in a greenhouse for 2 generations to control for maternal effects. And in 2021, we selected 4 populations from each region to study seed dormancy and seed viability differences in core and edge populations. For this 5-year study, we constructed sets of seed sleeves using chiffon fabric with 8 pockets to represent all populations in each sleeve. The seed sleeves are buried at 2 sites at Blue Oak Ranch Reserve, and each year a set will be removed from each site. Each set of seed sleeves contains 2,400 seeds. I predict that the dormancy rate will be lower in Dittrichia edge populations than in core populations, because over time, invasion velocity may have increased in the edge populations and result in reduced seed dormancy. As a companion experiment to this field study, I want to quantify seed germination rates, which cannot ethically be conducted in the field. I want to use seed from the same 2 regions to track germination in years 1, 2, 3, and 5, and parallel that with the field experiment. These two seed bank experiments will take a holistic plant perspective and feed into Sankey diagrams, which are a novel tool to track the fates of the seeds in a visual manner. Here we have the different fates of seeds terminating in decay, which is in gray or the letter A, in germination, which is green or the letter B, and in the retained seed pool in brown or the letter C, which is the proportion of seed that remains in the soil. Information about field experiment will help me understand the B and C portions, and the greenhouse experiment will help me model the differences between A and B. If I find that seeds that stay in the soil are viable after multiple years, it means that Dittrichia can wait for ideal cues to germinate. This scenario would imply that vegetation managers would need to continue checking in an area after they've removed Dittrichia in case there is live seed in that seed bank. However, if I find that germination is high and the remaining seeds are dead, then there won't be a long seed bank that managers need to deal with. So in summary, my proposed research investigates important ecological and evolutionary factors influencing the invasion of exotic species. This research has both conceptual and applied significance, furthering our understanding of weed management and supporting science-based risk assessment for Dittrichia graveolens. I will explore how disturbance, competition, and rapid evolution influence Dittrichia's ability to spread away from roadsides and compete with off-road vegetation. I will assess Dittrichia's potential impact directly through competition and indirectly through soil-mediated effects on other plant species. Finally, this research will contribute the first study to soil seed bank dynamics in Dittrichia, testing for evolutionary change in populations at the spreading edge and providing critical information for management. Taken together, this research will provide much needed information for management of a novel exotic species and advances research on how invasions arise and impact plant communities. With that, I want to thank and acknowledge my support system, my collaborators and funders. Thank you all again for coming, and I welcome your questions.
Thank you, Miranda. And we can either have people raise hands, or I think it will probably work out just fine to have people just unmute and ask their question, or if you want, you can put it in the chat.
Miranda, I have a question.
Yes.
I'm so curious, like what your plans are, because I never got to do anything like what you're planning to do with the seed banks with the greenhouse experiment. So like, what is your plan for like telling apart your germination from your decay? Are you going to be like pulling seed packets in the greenhouse at like some regular interval and checking to look for the see if you can tell one from the other if you're looking at like regular intervals?
Yeah, the plan right now is for in the greenhouse, rather than using seed packets the way we do in the field, that we'll just be able to, we'll have loose seed that is kept in a certain location within that pot. And after a rain event, then we'll go through and we'll pull things.
Awesome. I'm also curious, you mentioned when you were out in the field, you mentioned something about the patterns of invasion that you see. And I was just curious to know more about like what it is that you see and what it is that you think is going on and how that sort of fed into the decisions that you made for the experiments you're doing.
I grapple with the question of why is Dittrichia where it is? Why is it not where it isn't? And that's a really hard question to answer. It's hard to test for. So my research has led me to look at it from many different perspectives. And the patterns that I saw were patches of Dittrichia. And then some of the pictures that I've shown where you've got mowed vegetation, but then Dittrichia is growing up in it. So in some cases, there are patches of Dittrichia where there's nothing that seems to be able to grow underneath. And the question is that patch going to stay? Is anything able to come in after that Dittrichia, or does that patch change and morph in its size? And I have no way to have those answers at this time. I'm also not testing for those particular questions, but all of these things led me to really want to understand more about whether Dittrichia is inherently changing something about where it's living and able to suppress other vegetation in some way, or if there was some reason why it was relegated to the areas where it was living and unable to move further away to an area that, in my opinion, looks a much more favorable soil, better, water sources, and just that's where I'd want to live. So why is Dittrichia over there? So looking at those patterns of trying to understand why is it there and not over there led me here to try and kind of triangulate to get to that question, testing different ways of trying to figure out why.
Excellent.
Miranda, Rachel has a question about Dittrichia chemistry. Rachel, did you want to unmute and ask it yourself?
Sure. Yeah, I was just curious, does Dittrichia have secondary compounds? Are you thinking that there's something that it has that might be in the soil, or what are you looking for at that level?
There's research that has come out of kind of the Mediterranean area and Northern Africa area that suggests that in its home range, there are certain portions of Dittrichia that may have allelopathic tendencies towards just simple repression of germination. However, it seems to be variable depending on the season, when in the season you're collecting those plant tissues, as well as what parts of the plant you're collecting. So Dittrichia has a lot of chemical compounds. It's called stinkwort because I think it really stinks. Other people think it just smells like camphor or menthol or something nice and sweet. I think it really stinks, and those chemicals are going somewhere. And they're offending my nose, and I'm wondering, are they also doing something to that, to whether it's chemical warfare or changing something about the soils that is going to make it more challenging for other species to be recruited into those areas. So there's not a lot that I can really explore there. I started a few petri dishes of looking at it myself and didn't get very far with some lettuce seeds. So it just made sense to take a big step back and say, okay, so there's some literature out there. We know it's a really stinky plant. There's lots of chemical stuff going on. What's happening in general with the soils? Is it being changed? And I'll look to see as a first stage, can I find evidence for anything that is being changed in those soils? And if so, that leads me down a whole line of other questions that will come.
So you have papers on the identity of its chemicals? Have other people done the gas chromatography?
Yeah, I think there's definitely a paper out of China that did a chemical composition. There are certain cultures that are using the extracts as medicine. And so there has been a lot of identification of what these particular chemicals are, less clear as to how much they're actually doing an effect in the field and for, ecologically.
While I have the floor, I was also curious if anyone is looking at or has thought about, or if you thought about how the drought and the warming temperatures are play into the competition. And I realize this is like several jumps ahead of where you are with this, but maybe in its home range.
So Nikki, my collaborator who did study Dittrichia in the home range, found that it flowers earlier in the northern part of its home range where it has recently expanded. So certainly there has been a response to moving northward. In California, we're interested in finding out more about that, but certainly not in the purview of what I'm studying at this time. It'll be interesting to see how this field season, with it being so dry, will end up being for my competition plots.
Hi, Miranda. This is Laurel. It was very nice. I really liked the logical flow of the questions and the experiments that you did. I had a further question motivated a bit by Karen's about the seeds. And what's known about the seed dormancy in there? Because there's many species of plants that have a very deep dormancy, and it's really hard to break. And whether you can really parse that last step in that diagram that you were showing.
That last step in the diagram will be hard to parse. I plan to do a viability test. So far, evidence is suggesting that, I mean, you stick these seeds on in a petri dish on your windowsill, and they're going to germinate if you just give them a little bit of water. So there hasn't been a lot of evidence that they really are going to persist for years and years. And what I'm really trying to understand is, are we talking about 3 years? Are we talking about 5 years? Are we talking about more than 5 years? What is the management need for delivering resources back to that same patch where they pulled Dittrichia to understand after, you know, X number of years, they don't have to worry about that particular area anymore, and they can move on and start scouting for other areas where Dittrichia is gone. At that 5-year mark, we're hoping that, you know, it'll be more fuzzy, but, you know, at that point, we're hoping for the best and that those seeds won't be viable any longer.
Okay.
Hi, Miranda, it's John. I had a question related to, I like the balance of the greenhouse and the field component. A question I had about the field component is, I know among a lot of the other Asteraceae, seed predation is an issue. Also, the malformation of seed. Will you be looking at any of the seed in the field and for any kind of signs of damage or just inviable seed?
In the field, there's really no way for me to determine the demise of a seed. They're going to be in bags, so we're hoping that seed predation is not something that's going to occur by any sort of macroorganism. I can't rule out whether it died, whether it germinated and died, or, you know, pathogens came in. I would need more research would be required at that point. But really great question. I don't think that really there's been much explored about this particular species seeds. And so I feel like I'm scratching the surface. And as soon as I get some results back, that's going to just pour in lots of questions that need further exploration.
I think there's time for one more question. Mark, did you have a question?
If I can sneak it in. Hey, Miranda, you mentioned this, but I wanted to get straight. What's the rationale for dormancy to decline as you get to the outer edge of a species range?
Great question. The rationale is that when you are on, when a population is on an invading edge, it is advantageous to break that dormancy and not have long-term dormancy, but instead get that next generation going as fast as possible so you can move and keep spreading to a better location. So it's all about trying to find a better location for your offspring and to get away from whatever conditions you're in. So it would not be advantageous, I'm predicting, to have a long dormancy, because if you couldn't germinate that first year, odds are you won't be able to germinate the next year. So it would be better to, rather than wait to have a really robust plant that can produce tons of seeds and get out of there, make a really scruffy little plant and just get some seeds, move your genetics on down the line, find a new place to sow those seeds.
Yeah, I was going to say, I would have thought possibly the opposite, where as you get toward the center of a species range, where presumably environmental conditions are more appropriate, more consistently appropriate, dormancy might not be as important. Whereas if you're out at the outer edge of a species range, it's kind of moving into less appropriate habitats that, or less consistent appropriate habitat, that maybe dormancy might be more important to be able to persist at that point. It'll be interesting to see.
It will be. And yes, bed hedging at the edge also, there's viability in that argument as well, of, you know, reserve some seed because maybe next year would be better, or have reduced dormancy in your core range because you know that conditions are good. But if there are pressures in your core range for competition, maybe you're wanting to reserve some of that seed for better conditions next year. So it's, I think that both ways have strong arguments, but the theories that I'm considering here are well matched with how Dittrichia appears to move, and because it is not a perennial and it is an annual, it's got one shot to get those seeds out there, and then that's it.
Very
Well,
cool.
I think that's a great wrap up. And I want to say thank you to Miranda for a beautiful job on her talk. And for all of you for coming, thank you so much for coming. And I think a few of us are going to stay on and just have a little social check-in with Miranda, and anybody's welcome to stay for that. And but thank you.